Good morning, everyone. Today I will be talking about the journal club topic susceptibility of dental caries microcosm biofilms to photodynamic therapy mediated by photoenticin. It was published in 2021 in the Journal of Pharmaceutics with an impact factor of 5.5. I want to discuss some unique terminologies which will be helpful while discussing this journal club. We have to know about microcosm biofilm, a complex community of microorganisms grown in the lab that closely mimics the natural bacterial environment of the mouth. Photodynamic therapy, a treatment that uses light and a light activated chemical to kill harmful microbes. Photoenticin, a specific photosensitizer. It is a light activated compound used in photodynamic therapy to destroy bacteria in dental biofilms. Colony forming units, a measurement that counts how many viable bacteria are present based on their ability to grow into colonies. And the minimum biofilm eradication concentration, the lowest concentration of an antimicrobial agent needed to complete eliminate bacteria living in a biofilm. When we talk about the novelty, aims, and objectives, it is the first study to evaluate the photoenticin mediated photodynamic therapy against dental caries microcosm biofilms grown from human saliva. It assesses both the planktonic and the biofilm bacterial susceptibility to PDT, including karyogenic species. It combines microbiological and the structural biofilm analysis using confocal microscopy for a detailed evaluation. When we talk about its aim is to investigate the antimicrobial effectiveness of the photoenticin mediated PDT on dental caries microcosm biofilms. When we talk about the objectives, it determines the reduction in bacterial viability, evaluates the minimum biofilm eradication concentration, analyzes the biofilm structure and thickness changes using confocal laser scanning microscopy, and it compares PDT effects on planktonic versus biofilm bacterial forms. When we talk about the materials and methodology, the materials biofilm source, it is a stimulated human saliva from healthy donors to create microcosm biofilms. Photosensitizer, photoenticin at various concentrations, light source LED device emitting red light at 660 nanometer wavelength, microbial culture medium was MacBain artificial saliva medium for biofilm growth, and analytical tools were colony forming unit counting for bacterial viability. Confocal laser scanning microscopy was done for structural biofilm analysis. When we talk about the methodology, we collected the saliva samples from healthy volunteers and it was inoculated in MacBain medium to grow biofilms on glass cover slips. It was matured biofilms that were matured for 5 days before treatment, and it was divided into groups. First group was PDT with photoenticin plus LED light. Second group was photoenticin only, and third group was light only, and fourth was untreated control. Applied photoenticin to biofilms for 15 minutes pre-eradication, irradiated with LED light for 5 minutes at specific power density, collected biofilm and planktonic cells for CFU counting and calculated bacterial reduction, determined MBEC by testing different photoenticin concentrations, used CLSM to visualize biofilm thickness and live dead cell distribution after treatments. Statistical analysis test used was one-way ANOVA followed by Tukey's post hoc test for multiple group comparisons. Parameters compared were bacterial viability in planktonic versus biofilm forms, biofilm thickness and live dead cell ratio from CLSM, MBEC values for photoenticin against biofilm bacteria. Outcome: bacterial viability. Photodynamic therapy with photoenticine significantly reduced CFU counts in both planktonic and biofilm bacteria compared to controls. Biofilm structure: PDT group showed significantly thinner biofilms and higher proportions of dead cells than all other groups. MBEC, photoenticine mediated PDT, achieved complete biofilm eradication at a lower concentration than photoenticine group alone. When we talk about the results, when compared for planktonic bacteria, the group with the PDT and photoenticine caused a significant CFU reduction compared to all other groups. When talking about biofilm bacteria, PDT group showed the highest bacterial kill rate with CFU count significantly lower than light only, photoenticine only, and control groups. When we talk about MBEC, photoenticine mediated PDT eradicated biofilms at a much lower concentration than non irradiated photoenticine. When we talk about biofilm thickness, confocal microscopy revealed markedly thinner biofilms in the PDT group compared to other treatments. When we talk about the cell viability, live dead cell staining showed a predominance of dead cells in the PDT group while other groups retained mostly live bacteria. When we talk about the strengths of the study, use of human saliva derived microcosm biofilms closely mimicking the natural oral microbiome. Direct comparison was done between the photodynamic therapy, photosensitizer only group, light only group, and untreated control groups. Multiple outcome measures were done for microbiological assays, structural changes, and viability counts. Advanced imaging with CLSM provided detailed visualization of treatments' effects on biofilm architecture. Determination of the MBEC offered clinically relevant data on the minimum effective concentration for both biofilm eradication. When we talk about the limitations of the study, it was in vitro design which we may not fully replicate clinical conditions in the oral cavity. Short-term assessment without evaluating long-term biofilm regrowth or recolonization. Limited range of microorganisms. While microcosm biofilms are complex, they may not capture all the oral microbial diversity. Single photosensitizer, photoenticine tested, so results may not apply to other agents. Fixed irradiation parameters. No evaluation of the different light doses or exposure times for optimization. When talking about the study guidelines, it was an in vitro experimental laboratory study comparing the antimicrobial effects of the different treatment groups on dental caries microcosm biofilms. Guidelines compliance was arrived from the ARRIVE guidelines and in vitro experimental reporting standards. The guidelines that were not followed or partially followed were no formal sample size calculation or power analysis, donor variability not extensively characterized or controlled, blinding of analysis not mentioned, no reporting of reproducibility tests across different labs, limited discussion on potential confounding factors in the in vitro setup. When we talk about the future directions, conduct in vivo clinical trials to confirm efficacy and safety of the photoenticine mediated PDT in the real oral environments, evaluate long-term effects on the biofilm regrowth, recurrence, and microbial recolonization, test different photosensitizers and light sources to compare effectiveness and optimize protocols, explore combination therapies with remineralization agents for enhanced caries management, and assess patient-centered outcomes such as comfort, treatment acceptance, and cost effectiveness in clinical settings should be done. Thank you.